Namaste students. I am JVN Dr. Aziz Nand Khan from Department of Food and Biotechnology, Faculty of Agriculture and Veterinary Science, Jyoti Vidyapith Women's University Chapel. Today I am going to discuss about the topic monoclonal antibodies. What are the monoclonal antibodies and how the monoclonal antibodies are produced? It is the process of production of monoclonal antibodies. So first we need to discuss about the antibodies. What are the antibodies? Then we go to the monoclonal antibodies. Antibody is a protein which is used by the immune system to identify and neutralize foreign objects like bacteria, fungus, viruses, and each antibody recognizes a specific antigen unique to its target. So, they are very unique, they are very specific to a particular kind of antigen, a particular kind of pathogens. Okay, and their function is to neutralize and then identify. And on polyclonal antibodies are the antibodies that are derived from the different cell lines. Means these polyclonal antibodies can bind to different antigens. They are not specific to a particular kind of antigen. Now, monoclonal antibodies. The monoclonal antibodies are which are identical because they are produced by one type of immune cell, single type of cell, and all clones of a single parent cell. They are cloned. They are single type of immunoglobulins which are cloned structures, which are cloned antibodies. Isotype isotype are the according to difference in the heavy chain constant domain. Immunoglobulins are grouped into 5 classes, such as the IgG, IgA, IgM, IgT, and IgE. So they all are the isotypes. Now, talk deeply about the monoclonal antibodies. Monoclonal antibodies are the antibodies that are identical because they are produced by one type of immune cell. Single type of cells are used for their production. All clones of a single parent cells. And given any substances, it is possible to create monoclonal antibodies that specifically bind to any kind of substance, any particular kind of substance, and they can serve to detect and verify a substance. Uh, it is very important. The monoclonal antibodies are very important tool in the biochemistry, in the molecular biology, and the medicines. We have already discussed about the structure of the antibodies. So similar type of structures are observed in the monoclonal antibodies. And now discuss about the history of the monoclonal antibodies. The monoclonal antibodies history is very long. In 1890, von Behring and Kitasto discovered the serum of vaccinated person contain certain substances which are termed as antibodies. In 1900, Erich proposed the side chain theory. In 1955, Jen postulated natural selection theory, natural selection. Frank McLean Burnett expanded this natural and clonal selection theory, and then Porter isolated fragment of antigen binding and fragment crystalline from rabbit Y globulin. In 1964, Littlefield developed a way to isolate hybrid cells from two parent cell lines using the hypoxanthine aminopectin thymidine means HET medium, HET selection medium. In 1975, Kohler and Milstein provided the most outstanding proof of the clonal selection theory by fusion of normal and malignant cells, and this resulted in the first monoclonal antibody productions, and for which they were awarded as a Nobel Prize. So monoclonal antibodies are produced by a special technique which is known as the hybridoma. Means hybrid are created for the production of the monoclonal antibodies. So discuss about the hybridoma technology for the monoclonal antibody productions. It is a technique for the production of hybrid cell lines called the hybridomas. And by the fusion of what are the fused, what the two kind of cells are fused in hybridoma technology, antibody producing B cells are hybridized with the myeloma cells. And what is the function of the myeloma cells? Myeloma cells have the ability to divide, and they have the ability to grow in the tissue culture media. The hybridoma produce some antibodies which are produced that have single specificity and are called as the monoclonal antibodies. Means they can bind to a single type of antigens. So they are very specific. So that's why they are known as the monoclonal antibodies. These are the target based. These are produced by the target based or objective based. Okay, on they are produced because we have an objective to produce a single type of antibody. So that's why they are known as the monoclonal antibodies. And this technique was discovered by George Scholar of West Germany, Cesar Messing of Argentina, and Niels Jans of Denmark in 1975. And they were awarded for the Nobel Prize in 1994. And uh, if we talk about the principle of this theory, then this is based on the fusion between the myeloma cells, which are the malignant plasma cells, and the spleen of a suitable immunized animal. Spleen can produce antibodies, but they will die in short period. So we are using myeloma cells. We can take the use of the spleen, but these spleen will not survive in the culture media. So we are using myeloma cells. And myeloma cells are adapted to grow permanently in the cell culture. Myeloma cells cannot synthesize antibodies as they lack HGPRT gene required for the synthesis of the enzymes apoxanthin, guanosine, phosphoribosyltransferase or thymidine kinase. So they are not able to produce. And mutant myeloma cell cannot grow in the het medium. Only hybrid cell can grow. Okay? Only hybrid cell can grow. So HET medium is used for the screening of the hybrid cells. Only hybrid cell will grow on the het medium, hypoxanthine monotherine medium. And the hybrid cell will grow in the het medium, and they will produce monoclonal antibodies. Now discuss about the method of the production of immunoglobulin. First step is the immunization of any. If we insert a antigen into a mouse, then this process is known as the immunization. Okay, and the antigen is very specific here. This antigen is the antigen for which we want an antibody. This is a specific antigen. So first, what we do, we insert the injection of a particular antigen into the mouse, and the mouse gets immunized. Okay, the mouse will get immunized, and after immunization, the mouse will produce antibodies. Okay, they will produce antibodies, And as we know that mouse is also a mammalian, so they have spleen. So the antibodies which are produced by the mouse immune system will be stored in the uh, spleen. So we isolate the spleen, and then we isolate the B cells from the spleen. Then we cultivate the myeloma cells in the culture medium, and we fuse the B cells and the myeloma cells. The B cells which were isolated from the spleen of the mouse, and the myeloma cells are fused on the medium. When they will grow, when they will grow, we will separate the cell lines, and we we need to screen these cell lines on the HED medium. And we know we have already discussed the principle. Only the hybrid cells will grow on the HET medium, het medium. And then when they will grow, we will screen them. We will screen them, and we conduct the in vitro and in vitro methods for the multiplication of the uh, hybrid cells. After that, when they will grow, we will harvest the cells, and we perform the production of the monoclonal antibody from these hybrid cells. And first, immunization a rabbit through repeated injection of a specific antigen for the production of specific antibody facilitated due to proliferation of the desired B cell. This will produce tumors in the mouse or a rabbit. And then we culture separately the spleen cells that produce specific antibodies and the myeloma cells that produce tumors. Then for the fusion of spleen and the myeloma cell is induced by the polyethylene. If we were talking about the fusion, so how the fusion is carried out, the fusion is carried out by the use of the polyethylene glycol, and this will produce the hybrid cells. These hybrid cells are then grown in the selective medium, which is hypoxanthine, aminopterin, thymidine, het medium. Het medium contains a drug which is known as the aminopterin that blocks the one pathway of nucleotide synthesis, making the cells dependent on another pathway that needs HDPRT enzymes, which is absent in the myeloma cells. So myeloma cells will not only grow, they will allow the B cells to grow. And the myeloma cells that do not fuse with the B cells will die. So only only the hybrid cells will grow on HED medium. B cells that do not fuse with the will also die because they lack tumor tumorigenic property of immortal growth. So undesired cells means unbound B cells and unbound myeloma cells will die. And after that, we go for the selection and so stories. We select the desired myeloma cells for the cloning and antibody production, and then we will prepare the single cell clones that will go on and then screen for the antibody production of the hybridoma. Only one in several hundred cell hybrids will produce the antibodies of the desired specificity, and then we will culture selected hybridoma cells for the production of monoclonal antibodies in large quantities. And hybridoma cells can be frozen for future uses or can be injected in the body of an animal so that the antibodies will be produced in the body and recovered later from the body fluid. So this is all about the production of monoclonal antibody. So students will be able to understand what are the monoclonal antibody, what are the polyclonal antibody, what is the difference between the polyclonal and the monoclonal. General introduction of monoclonal antibody and what is the method of the production of monoclonal antibody, what is the principle of the production of monoclonal antibody, and which kind of questions can be asked here. Uh, the examiner can ask that explain monoclonal antibody and their productions, explain the principle of production of monoclonal antibody, explain the application of monoclonal antibody. So I can also explain the production, explain the application of the monoclonal antibody. Uh, these monoclonal antibodies are present in the biochemistry experiments also. So this is the application of the monoclonal antibody. And the one example is that they are used in the pregnancy test. These monoclonal antibodies are also used in the pregnancy test. They are also used in the ELISA test. They are also used in the diagnosis of disease. So these are the applications of the monoclonal. So my lecture is now over here. If you have any questions, you can ask me. Thank you very much.